Hi everybody, I'm Dr. Mona Okal, Assistant Professor of Biopharmaceutics. Today I will give you a lecture about the biopharmaceutical development and manufacturing of biosimilar, and this is part 1, and this is the 10th lecture, and we will discuss many aspects regarding how to develop and how to set a good strategy for manufacturing of biosimilar. So in a previous lecture, we talked about the business, health economic, and intellectual property landscape for biosimilar. Today, we will get started and talk about biosimilars, drug substance development and manufacturing, effective CMSC strategy. So first of all, we need to know what's the meaning of strategy? Strategy generally, it's a course of activities that lead to defined objectives. So when we talk about CMC strategy, first we need to have CMC stand for what? C is stand for chemistry, M for manufacturing, and C for control. So all together, chemistry, manufacturing, and control, CMC. So for an effective CMC strategy, that means there is a course of CMC activities that lead to defined regulatory objectives, such as initiating a phase 1 in human clinical studies, initiating phase 3 by vital clinical studies, and obtaining market approval. So when we define a CMC regulatory strategy, it is always a first step for any drug. But when we talk about a biological drug, a biosimilar drug, it's not an easy things like for the chemical drug. It's a challenging step. So for any chemical drugs, it's chemistry, manufacturing, and control, CMC. So in the coming slide, I will not keep saying chemistry, manufacturing, controls. You need to have to know from now CMC what it stands for. So for any drugs, chemical drugs, its CMC section contains a details aspect about that chemical drug, including chemical properties of this chemical drug, about its manufacturing process, how we can do development for this drug, and how the process for the production, also about the control mechanism including mainly about the analytical testing that occurs in both process and of the finished drug. This is so important to show that the process of drug development and manufacturing is under control, and it is in the same time reproducible. Here when we talk about the effective CMC strategy for the candidate biosimilar, the first point regarding the chemical properties, it is much easier for the candidate biosimilars because the properties, it is already understood about the chemistry and the properties of the drug. Why? Because of the thorough characterization of the reference drug. As we know that both the similar drugs and the biological drug are much related and closely related in the chemical structure. But about when we talk about manufacturing process, it's much challenging process because we should keep all the time the biosimilarity between the biosimilar drugs and the reference drugs, which means, in other words, that the biosimilars it should closely match the reference drug product, and in the same time should demonstrate this biosimilarity along with a strong analytical package, as we discussed before in the previous chapter about the studies for the similar drugs and biological drugs. The difference is about the biological drugs. Most of the studies extensively are about the clinical studies, while for the biosimilar drugs, most studies are about the analytical aspect in order to approve all the time that the similars are closely related to the biological drug. So the first step to obtain an effective biosimilar CMC strategy is to understand extensively the reference drug. When we say understanding the reference drug, that means we need to know many aspects about the reference drugs. First things, we need to know the biochemical properties of the reference or innovator drug product. But in the same time, the biosimilar developer doesn't have the access to any proprietary information about the reference drug, including the development reports. They don't have access for it, batch records, release specification for the drug substance and product. So what do you think the solution, or how they can get access, or what kind of information that the biosimilar developer can get about the reference product? So the biosimilar developer can get some information about the biologic reference. How? By different public sources. As it said, public sources, that it's accessible to all the people. It's not private for the company. This is this access from where? From publication, as many the biological innovators, they do many publication to approve the indication of their drugs, or the importance of their biologics, so they can get some information about the publication. Also from the presentation or information that are accessible from regulatory agencies, and also from certificate of analysis, which contain much information about the biologic drugs, but still there's some information they are not accessible to the biosimilar developer. So we come up with a new definition. It is the critical quality attributes, CQAS. So those with background, and they have a good background in the development of similar types of a protein product, they can help to identify basic aspects of the drug, such as amino acid sequence, type of the product like Ig antibody, fusion protein, and enzyme. So along with this information, they can know also about the mechanism of action and dosage and formulation. So they need expertise to help them to find out how they can develop the biosimilar in a way that these biosimilars are closely related to the basic aspect of the biologic drugs. So the critical quality attributes for the biosimilar drug much closely match that for the reference product. So this is the key test of biosimilarity. That means this stipulates avoiding having any clinically meaningful differences of biosimilar drugs from the reference product, and this is guarantee that this biosimilar drug is interchangeable with the biological drugs. Also, parts of understanding the reference drug, that the critical quality attributes could represent, as we said before, it could represent the protein functional aspect like antibody binding to its target, and this is very important for the biological activity of the biosimilar. Also, it can contain information about the enzyme activity that helps to determine the mechanism of action of the protein. Most importantly, to talk about is about the post-transitional modifications like glycosylation, and this is why important, because it affects the secondary aspect like effector function, as well as the pharmacokinetic and pharmacodynamic profile. At the end, all together will affect the biological activity of the biosimilar drug. Moreover, the critical quality attributes could represent and include information about the protein molecular state, such as aggregation or degradation. And this is very important, and it has a special concern for protein therapeutics. Why? Because this is very important when we talk about immunogenicity. We know, as we know, and we discussed before, that aggregation or degradation, that they have the potential to cause immunogenicity or provoke the immune system. So this is very important to be part of the strategy, CMC strategy for the biosimilar drugs, that we have information about the aggregation or degradation, because for protein therapeutics, presence of aggregation or degradation, they have a higher immunogenicity. Yeah, and this is not acceptable and undesirable things that we want to have with the biosimilar development. And also, we could know from the molecular state of the protein, the secondary, the tertiary, or the higher order structure. Why? Again, it's important because this is will have impact on biological activity of the biosimilar. And keep in your mind, in all the steps of the biosimilar development and production, biosimilarity should be attainable at all the time. And this imposes to have many analytical studies to improve all the time this fact, and this will add also cost for biosimilar, and this is justify also why biosimilar is still expensive, but still they are cheaper compared to the biological drug. Also, similar to biologic product, a biosimilar product should be evaluated for a process-related residual impurities, impurities like come up from host cell protein, which abbreviated like HCP, host cell DNA, HD, HC, DNA, and residual protein A, when protein A chromatography is used in the process. A protein A chromatography, it is used for antibody purification, and it relies on a specific and reversible binding of antibody to an immobilized protein A ligand. Protein A, usually it is 56 kilodalton surface protein of Staphylococcus aureus, and protein A chromatography, usually it is well known because it is very robust purification procedures, and it's used as a capture step due to its specificity. So they use many techniques in order to detect this is impurities, and this is undesirable impurities, like they use the ELISA to, not ELISA, they used ELISA to detect HCB. Again, HCB stands for host cell proteins and protein A purities. They use quantitative PCR to measure the DNA, and they use generally the general safety assay like endotoxins, which we will discuss later. It comes up from the animal raw materials, and also they check for the sterility. So, it is well known for any therapeutic protein. Um, they have a family of closely related variants. Variant, it means that they have the same amino acid sequence, but they vary in the level of modification, either from variation within the post-transitional modification, or the variation result due to the degradation, such as deamidation and oxidation, which we discussed this both types of chemical instabilities before. So these variants, they have comparable activities like target binding, but they vary in terms of what? In terms of the effector function or pharmacodynamic and pharmacokinetic profiles for these variants. So, according to the biosimilar developer, we we have to know the allowed range of these variants. How? By doing extensive testing for many lots or lots to lots of the reference product. That will help to reveal and uncover the extent of variation for each critical quality attribute. But here, when we say that we want to extensively testing many lots of reference product, how many lots of that reference product should be tested in order to know the allowed range of these variants? Recent approvals of some biosimilars what suggest it should be at least 20 lots should be assayed, and with more being better in terms of range setting. So the minimum is 20, more, let's say it in Arabic, خير وبركة. Yeah, so recent approval, so suggest 20 lots of the reference product should be tested in order to know, tested for what? Tested for the variant in order to know the allowed range of these variant. So why it's important to testing the originator lots? The importance of testing of the originator lots in revealing the considerable differences in one or more CQS defining groups of lots with different profile. So this is important. Why? Because it indicates that the innovator conducted a process changing. They assessed comparability before and after the change, and they obtain approval by the regulatory agencies for the new process product. As we know that as these biological drugs, they are produced from living cell, they have variation from time to time. So all the time, even the biological drug should be tested that these variation will not affect the identity of or the function of this biological drug. So lots aged for varying length of time under the indicated storage condition will reveal the stability parameter. It means that we need to get some lots of the reference drug that they newly produce after 6 months, after different time, yeah, in order to see to reveal the stability parameters for the reference drug. How can what is the stability for this drug? How it can stand for how many time, for how long time it can be still stable. So we discussed the first step to obtain an effective CMC strategy is to know and understand the reference drug. The second step we should take it in our consideration in order to obtain an effective CMC strategy is to know the expression system and the clonal selection for the biosimilar drug. So the biological expression system for the production of recombinant protein therapeutic ranging could be simple from the simplest one from microbial system to complex one from the mammalian cell lines. So if we want to start with the simplest microbial cells such as E. coli, E. coli can only express protein, as we know, without post-transitional modification like glycosylation. They are producing effectively the protein but without any post-transitional modification. So any simpler protein that lack this modification bacterial system can be attractive choice due to their rapid growth rate and productivity. So if the protein of interest that we want to produce doesn't have or we don't want to make a glycosylation for this protein, so the bacterial system, it is like a system of choice and it's attractive because rapid growth rate and productivity. But So the protein produced from the bacterial system, which lacks the post-translational modification, also it presents a challenge in terms of the recovery because it's produced in a reduced and unfolded form and including in bodies. So we need to have another bacterial system that can secrete the folded protein such as the bacterial system Corynebacterium Ajinomoto. But again, the protein here still lack the post-transitional modification. If we want to move to a higher order microbes, which can produce protein with the post-transitional modification, the expression system of choice is the yeast expression system, like P. pasteurus and S. cerevisiae. Both produce some post-transitional modification, but still in a more primitive form, such as high mannose glycosylation. So, if we want a ready secretion of recombinant protein that have a full post-translational modification, the mammalian cell lines such as CHO, and PER-C6 have been expression system of choice. NS0, this is a cell line that are a model cell line um, derived from non-secreting imurine myeloma used in biomedical research and commercially in the production of therapeutic protein. While the bird C6, it is a cell line that produce from or derived from human embryonic retinal cells, originally from the retinal tissue of 18-week-old fetus that aborted in 1985. Then they further developed and prepared it as a cell line. How? By transfection with a defined E1 region of the adenovirus type 5, followed by selection of transfectant with an immortal phenotype. Immortal phenotype, it means that they will continue replicate without death. So this is why important, because it's used to produce a variety of biopharmaceutical products, including vaccine, gene therapy products, and antibodies, also in other therapeutic protein. So occasionally, an alternative expression system, such as one based on insects and insect cells, or even green plant, is utilized for specialty product such as vaccine. This figure explains glycosylation in different system, yeast, insects, and the plant, and which is very important and critical for attaining biosimilarity. The details inside this figure are not included in the exam, just for your knowledge. Also, it's worthy to mention that there could be an economic advantages for production of simple proteins that lacking the post-translational modification which produced from the bacterial expression system due to the productivity and rapid growth rate. However, if we going to a high-yielding microbial system such as mammalian cell, we should pay attention for the differences that could be produced in some post-transitional modification between different type of the cell, such as sialic cell, sialic acid added in NANA and NGNA, or type of linkage between sugars. In NANA and NGNA, they are both glycoproteins, and the most common sialic member found in glycoprotein. It is the Nu5AC, which is correspond for NANA. But NANA is found in both human and non-human cell, while LNG-NA it is not found in human, and it is hydroxylated derivative of NANA. And since it is not found in human cells, so this is considered an immunogenic, and attention should be paid for this kind of post-translational modification. So the key focus and success for biosimilar developer is choosing a comparable system that used for the reference product, which means that we want expression system that produce both transitional modification as close as possible to that produced in the reference product. To fulfill these things, the safest approach is to use the same type of expression system. Even sometimes it's not the same type also, it's the same specific strain of mammalian cell, like the example shown here, that used for the reference. Also, it should be mentioned that the biosimilar developers take in their consideration in selecting the clones for production of recombinant protein is the productivity of the expression system. Usually, they are seeking the one with the highest productivity. And when we talk about productivity, we're talking about the function of both the specific expression rate, which is expressed in picogram per cell per day. Pico is 10 to power minus 12 of the gram, and also the variable cell density, how much cell are exist in a specific volume and they are still alive. So ideally, they are seeking a system with highest productivity that can have high expression rate and high variable cell density. So also here I'd like to add just one more information that we talk about transduction, and sometimes it come up with us a term transfection. One simple difference between transfection and transduction, that transfection is a process of introducing nucleic acid to cells into the cells by non-viral method, like using naked DNA or naked or plasmid DNA, while transduction is a process whereby a foreign DNA is introduced into another cell via a viral vector. In addition, in development of biosimilar, however, a new parameters into the selection process, which is how closely does the product from any given producing a clone match that of the reference product. So while this is mostly a function of both transitional modification, it may also be influenced by potential degradative processes in some clones, such as oxidation and the amidation. So this new criterion supersedes the productivity in terms of importance. Why? Since any biosimilar structural features must match as closely as possible to the reference product. So therefore, some highly productive clone may need to be passed over in favor of less productive ones that produce a protein more comparable to that of the reference product. Also, one of important aspects is the development of clones for the production of recombinant protein. They requiring that to have clonal stability. When we talk about clonal stability, that needs to be assessed how by repeatedly passaging the cells, typically for 20 passages or 50 generation, and measuring productivity and protein quality at selected time point during these passages. To explain this in more details, let's say like in my PhD project, I use many immortal human cancer cell lines. The first time I bought this one from the company, it comes up with a zero passage. Passage, it means that when I growing these cells for the first time, then I want, and they cover all the surface of the flask, and I want to detach these cells from the surface by adding a trypsin. They will detached. Then after make a suspension, I want to take half of the amount, or depend on the dilution that you want, or you want your cells to be ready for culturing again. Okay, then I take, let's say, make dilution 1 to 10 of these cells. Then I return the one part of the cells to the flask again and put it in the incubator. Here, what I should write, I should write P number 1. P1, also beside the name of the cell lines, it means passaging number 1. Let's say I do again detaching the cell again, it will give another number, page B2, its passage number 2, 3, 4, 5, etc. This is should be assessed when the cells reach 20 passage or 50 generation. Usually, it's recommended not to continue your research when the passage number exceeds 50. Why? Because some of the cell line, and even myself, I face this situation that the protein production and the quality it differs. It could be it could decrease, could be increased, could have post-transitional modification. So when we talk about biosimilar, we talk about protein. So we need to check this every time when passaging the cell. So checking clonal stability, it's so important, as I said before, because they may lose their ability to express protein or alter their expression over time. One of the cell lines I dealt with, that after exceeding the 50 passages, it didn't produce anymore the protein of interest that I'm looking for. So this is an example how they can lose their ability to express a protein or alter their expression over time. So this is need to be eliminated from the consideration when we talk about biosimilar, because we want to guarantee and make sure that biosimilarity is attainable at all the time. So with this consideration, should be recommended? It should be recommended to select a primary clone and at least 2 backups for the clonal stability study. So as I said, I'd like to always give you an example of my study, because if I want to guarantee that still I have stock of these cell lines at a lower passaging number. As I told you for a while, that when I make detaching the cell, I make dilution like 1 to 10. 1 to 10, it means if the total volume is 10 ml of these cell lines, I take 1 ml, put it in the flask, and add 9 ml of the cultural media. So the 9 ml remaining, I can discard it, or I can centrifuge it, and the compact amount of the drugs of the cells, I can freeze it. So I have a stock of the this cell line at lower passaging number. Also, the product expressed by all selected clones should be evaluated and pass all the criteria that are identified by the critical quality attributes for the full culture period. So after understanding the reference drug, then selecting the right expression system, we need to know more information about the development of upstream and downstream processes and scale up to manufacturing for biosimilar. Note that biosimilar development follows the same path as a new biopharmaceutical development in terms of upstream. When it's upstream, it means culture, and downstream purification processes development. So after selection, as I said, of the suitable clone to produce biosimilar product, the next goal and the next step is to produce the protein that with only traces of levels of impurities via developing consistent and high yielding processes, because impurities are undesirable, because we want the biosimilars to be as close as possible to the reference product. So the upstream development is typically carried out, as we discussed before a month ago, in bioreactors, which range from multiple mini bioreactors, which shown in the next slide, or like benchtop bioreactor. So this is the pictures for the one with 15 ml and the one with the 250 ml mini bioreactor. So we can make a varying, we can vary the culture media, feeds, bioreactor parameters to produce the most protein in a shortest time. So, while for the downstream, the purification step, the approach is to use a system of filters, chromatographic columns in order to purify protein from impurities, as it's discussed in details also in the first exam material, by either binding, retaining it while impurities can pass through it, or vice versa. So we do this. Why? To ensure that the selected conditions produce protein highly similar to the reference product. Keep in your mind, biosimilarity, we want it to be attainable at all the time. It's also it's worthy to be mentioned that some downstream processes, purification steps, have the potential to induce such structural changes in the protein. For example, deamidation has the potential to produce acidic variants that may pull the protein outside the isoelectric point range of the reference product. The isoelectric point, this is called sometimes pI of the protein, is the pH at which the net charge of the protein is zero. So for protein which with many basic amino acids, the pI will be high, while for the acidic protein, the pI will be lower. So the newer charge based techniques help much in this case, is because it is possible now to separate this charge variant, which in turn helps in removing the undesirable species in terms of biosimilarity. So these approaches, it will add complexity in producing or in the production of biosimilar, and add more steps and processing steps compared to that produce similar to the reference product. So, therefore, it is important to know that the consistency and the robustness in the production and the purification of biosimilar is such an important goal, like for any biopharmaceutical product. Also, additional steps, as we said in the previous slide, are added in the downstream processes in order to get rid of this variant. However, these additional steps also have the potential to cause the following. So when we selectively removing the undesirable charge variant in the purification process via specialized technique like charge displacement or cation exchange chromatography, this what can cause? This can cause a single charge change on the large protein that will result the new protein differs from that of the reference drug. Also, these additional steps can cause and has the potential to cause reducing in the yield of the protein significantly. Why? Because we need to closely fractionate this similar species. So again, the crucial steps in the production of biosimilar is to select cell line and growth conditions that result in the production of a protein that high similar profile to that of the reference product. And again and again, we want to make sure that the biosimilarity is attainable at all the time. So, in this approach, these approaches yield higher purity in terms of lower residual hostile proteins or lower level of aggregation or product-related integrants, as we do additional steps for purification. That it is helpful and beneficial since the components of the reference product are these components are undesirable. So we need to do finally a full viral clearance study, and this is required for biosimilar downstream processes such as for any biopharmaceutical new biopharmaceutics. So these viral clearance studies are mandatory for pharmaceutical production processes where material of a human or animal origin is included. So the objectives of these virus studies is firstly to define the processes step that may inactivate or remove viruses. Secondly, to estimate the overall virus reduction obtained by manufacturing process. So indeed, a bioprocessing techniques improve, increasing lower levels of such impurities are expected and may will be that the reference product has already seen several process improvement over its lifetime to produce a purer material. Also, we discussed the development of upstream, downstream processes. Now we will talk about the scale-up of production for the biosimilars. First, scale-up, it means increasing the batch size. So the way or the path of scaling up for the biosimilars, it's like for any biopharmaceuticals except one step. The one step we I mentioned it many times before that we need to make or it stipulates a constant assessing for the effect of this scale up on the similarity of biosimilars to the reference product. So many factors and effects in the production of biosimilars, it needed to be assessed continuously over the range of a bioreactor during the scale up. First, we need always to assess the level of oxygenation, carbon dioxide, pH, and the nutrient waste product, as well as the rate and efficiency of mixing inside bioreactor, because all of these factors will affect the biosimilarity of the protein being produced. Also, to keep in your mind that the product quality should be assessed along the way, whether following initial small-scale bioreactor or large bioreactor. All of these factors should be assessed all the time, and biosimilarity should be also being assessed. So, regarding product process qualification, process characterization, and validation, these areas of development are so similar to those that needed for novel biopharmaceuticals. However, the steps for biosimilars are much easier than the any other novel biopharmaceuticals. Why? Because already we have the knowledge, and even the knowledge is more advanced because we learn a lot of this information from the reference product. Also, for the control strategies, this is particularly strict for biosimilar. Why? Because as we say it I said it many times and I will keep saying that the need to maintain biosimilarity to the reference product at each step of the production and purification. Hopefully that this presentation helps you to understand and to get more information about biosimilars, and we will continue explaining and more information about biosimilar. Thank you for listening, and have a nice holiday.